The overall goal of the following experiment is to obtain a transcriptome-wide map of the binding sites of an RNA-binding protein at individual nucleotide resolution. This is achieved by UV irradiation of living cells to covalently cross-link proteins and RNA in vivo. As a second step, the RNA-binding protein is immunopurified under stringent conditions in order to recover the cross-linked RNA fragments. Next, reverse transcription generates cDNAs that often truncate at the attached peptide, which preserves the information on the cross-link position within the RNA. Results are obtained that show all binding sites within the transcriptome based on high-throughput sequencing of the cDNA library.
This method can help answer key questions in RNA biology, such as how the interplay of different RNA-binding proteins regulates RNA processing.
The main advantage of this technique is that we increase both the performance and the resolution of our transcriptome-wide maps of protein-RNA interactions.
To begin the procedure of UV cross-linking tissue culture cells, remove the medium from a 10 cm plate of HeLa cells, add 6 ml of ice-cold PBS and place the plates on ice. One plate is sufficient for 3 experiments. Remove the lid and irradiate the cells once at 254 nanometers with 150 millijoules per centimeter squared. Harvest the cells by scraping them off the plates with a cell lifter. Transfer 2 ml of cell suspension into each of 3 microtubes. To pellet the cells, spin them at top speed for 10 seconds at 4 degrees Celsius. Then, remove the supernatant. Snap freeze the cell pellets on dry ice and store at -80 degrees Celsius until use. Add 100 microliters of protein-A dynabeads per experiment to a fresh microtube. If working with mouse or goat antibodies, use protein-G dynabeads. Wash the beads twice with lysis buffer, which can be found in the accompanying written protocol. Resuspend the beads in 100 microliters of lysis buffer with 2 to 10 micrograms of antibody. Rotate the tube at room temperature for 30 to 60 minutes. Then, wash the beads 3 times with 900 microliters of lysis buffer and leave them in the last wash until you are ready to proceed to immunoprecipitation. Thaw the cell pellet on ice, resuspend it in 1 ml of lysis buffer and transfer it to a 1.5 ml microtube. Prepare a 1 to 500 dilution of RNase 1, then add 10 microliters together with 2 microliters of turbo-DNase to the cell lysate. Incubate the samples for exactly 3 minutes at 37 degrees Celsius, shaking at 1100 rotations per minute. Immediately transfer to ice. Then, spin the samples at 4 degrees Celsius and 22,000 times gravity for 20 minutes to clear the lysate. Carefully collect the supernatant, leaving about 50 microliters of lysate with the pellet. To immunoprecipitate the beads, remove the wash buffer, then add the prepared cell lysates. Rotate the samples for 2 hours at 4 degrees Celsius. Discard the supernatant and wash the beads twice with 900 microliters of high salt buffer, which can be found in the accompanying written protocol. Finally, wash the beads twice with 900 microliters of wash buffer, then proceed to dephosphorylate and add a linker to the RNA 3' ends. To label the RNA 5' ends, working behind a plexiglass shield, remove the supernatant from the last wash of the linker ligation reaction and resuspend the beads in 8 microliters of hot PNK mix. Incubate for 5 minutes at 37 degrees Celsius. Remove the hot PNK mix and resuspend the beads in 20 microliters of 1 times new page loading buffer. Then, incubate the sample on a thermomixer at 70 degrees Celsius for 10 minutes. Immediately place the sample on a magnet to precipitate the empty beads. Load the samples onto a 4 to 12% new page bis-tris gel according to the manufacturer's instructions. Also load 5 microliters of a pre-stained protein size marker. Run the gel for 50 minutes at 180 volts. Transfer the protein-RNA complexes from the gel to a nitrocellulose membrane using the Novex wet transfer apparatus according to the manufacturer's instructions. After the transfer, rinse the membrane in PBS buffer, then wrap it in Saran wrap and expose it to film at -80 degrees Celsius. Perform exposures for 30 minutes, 1 hour, and overnight. Next, isolate the membrane region according to the protein-RNA complexes from the low RNase experiment using the autoradiograph as a mask. Recover the RNA from the membrane with proteinase K digestion and phenol chloroform extraction. Reverse transcribe the RNA using one of the iCLIP primers. To gel purify the cDNA, mix 6 microliters of cDNA sample with 6 microliters of 2 times TBE urea loading buffer. Just before loading the samples onto a gel, heat the samples to 80 degrees Celsius for 3 minutes. Then, load the samples onto a precast 6% TBE urea gel along with a low molecular weight marker. Allow the gel to run for 40 minutes at 180 volts as described by the manufacturer. Using the upper die and the marks on the plastic gel support to guide excision, cut 3 bands at 120 to 200 nucleotides, 85 to 120 nucleotides, and 70 to 85 nucleotides. Note that the iCLIP primer and the L3 sequence together account for 52 nucleotides of the CLIP sequence. Add 400 microliters of TE and crush the gel slice into small pieces using a 1 ml syringe plunger. Incubate the gel pieces with shaking at 1100 rotations per minute for 2 hours at 37 degrees Celsius. Place 2 1 cm glass pre-filters into a CoStar SpinX column. Transfer the liquid portion of the sample to the column. Spin for 1 minute at 13,000 rotations per minute into a 1.5 ml tube. Add 0.5 microliters of glycoblue and 40 microliters of sodium acetate, pH 5.5, then mix the sample. Add 1 milliliter of 100% ethanol, mix again, and precipitate the sample overnight at -20 degrees Celsius. Finally, circularize the cDNA molecules to attach the adapter region to the 5' end. Next, relinearize and PCR amplify according to the accompanying written protocol. Prior to the sequencing of the iCLIP library, the success of the experiment can be monitored at 2 steps: the autoradiograph of the protein-RNA complex after membrane transfer, and the gel image of the PCR products. In the autoradiograph of the low RNA samples, diffuse radioactivity should be seen above the molecular weight of the protein. For high RNA samples, this radioactivity is focused closer to the molecular weight of the protein. When no antibody is used in the immunoprecipitation, no signal should be detected. Further important controls for specificity of the immunoprecipitation either emit UV irradiation or use cells that do not express the protein of interest. The gel image of the PCR products should demonstrate a size range that corresponds to the cDNA fraction. Note that the PCR primers P3 Celexa and P5 Celexa introduce an additional 76 nucleotides to the size of the cDNA. If no antibody is used during the immunoprecipitation, no corresponding PCR product should be detected.
While attempting this procedure, it is important to remember that each of the 64 steps is critical and has to be performed with 100% accuracy.
iCLIP data can be computationally integrated with transcriptome-wide functional assays. This can, for example, gain insights into the position-dependent regulation of RNA processing. For computational data analysis, we would like to refer to our recent publications in collaboration with Nicholas Combe at the European Bioinformatics Institute and Blaž Zupan at the University of Ljubljana.
And now it's time that you start your own experiments. Have fun!